Well, good morning, everyone, and welcome to Garvan's virtual public seminar. Today's topic is diseases that affect us most, and this is the first in a two-part series. For those of you who don't know me, my name's Mara Jean Tilley. I have the great privilege of heading up the Garvan Research Foundation, which is the marketing and fundraising arm of the Garvan Institute of Medical Research. We're in for a bit of a treat this morning. We've got four absolutely phenomenal young, bright superstars, and we're rejoined by Professor Chris Goodnow, Garvan's Executive Director, returned from his well-deserved couple of days off. As many of you will know, he was an apology for the last one. Chris, great to have you back again with us this morning. Before we begin, I would like to pay my respects to both the Gadigal and Gundagara peoples, the traditional owners and custodians of the lands on which the Garvan Institute and our Moss Vale facility are located. I personally pay my deep respects to the traditional owners and would like to recognise the continuing connection to land, waters, culture, and pay my respects to elders past, present, and emerging indeed. A little bit of quick housekeeping for those of you who are new to the virtual seminar experience. We will be having a Q&A in the latter half of the seminar. So if you look at the bottom of your screen, you'll see that there's a Q&A little box there. If you click on that box, you can type in any questions that you have as they come to mind, and we'll do our very best to get to those, to get to as many of those questions as we can before we conclude today's session. We have a lot of new faces virtually in the audience today. Welcome. We're also joined by a number of our Garvan, long-standing Garvan supporters, our donors, our partners for the future, our board directors, our volunteers. Thank you so much to each and every one of you. And to the newcomers, we really hope you enjoy today's session. I would like to shout out a quick acknowledgement to Garvan staff. So as many of you will know, we've got more than 700 people working across four major themes of research, and everybody's been doing a phenomenal job under difficult circumstances. And particularly I shout out to our clinician scientists who are juggling clinical responsibilities at a particularly difficult time with COVID-19 and also driving their research efforts forward. So a big shout out to all of you. I'm going to hand over in just a moment to Chris Goodnow, Professor Chris Goodnow. Before I do, just a quick introduction for Chris. So Chris is truly a world-renowned immunologist, and he joined Garvan in 2015 as Deputy Director before taking over as the Executive Director in 2018, and he's just an incredible leader for all of us here at Garvan. Chris is dedicated to understanding the immune system, autoimmunity and immune deficiencies, and he's best known for integrating molecular genetics and genomics with immunology. He's currently leading the HOPE research program, which is a transformative research program looking at more than 40 autoimmune diseases. This is a fantastic initiative of the Garvan-Weizmann Partnership and is very generously supported by the Bill and Patricia Ritchie Foundation. So please join me in welcoming Professor Chris Goodnow.
Thanks, Mara Jean, and welcome and thank you to joining today's seminar. We've got a really interesting lineup of speakers, so I think you'll enjoy it. They're only going to speak for very short amounts of time, but that allows lots of time for people to ask questions, and I know you'll have them. Look, 2020 has been a challenging year in Australia and around the world. You all know that, and certainly for Garvan, it started and continues with a bang. You know, our goal at Garvan is to use all the information that's encoded in our genome to better predict and diagnose and prevent major diseases that affect our society. And our researchers, as we're going to hear about today, work across four intersecting research themes. At that very core, that enabling transformative technologies of genomics, epigenetics, cellular genomics, and you'll hear that woven into the talks today. Cancer, of course, always a major issue, our biggest single theme. Diseases of immunity and inflammation, and diseases of aging, particularly the ones that affect our bones, which is what we'll hear about today, brain, metabolism. So that diverse breadth of research is it nevertheless overlaps quite spectacularly, and that's part of what makes Garvan special, especially when you put it together with genomics that breaks down the barriers. Now that depth and integration also meant that when COVID-19 started spreading around the world in January, we all looked at that and said, look, on top of everything we're already doing, we need to do even more, because we have a lot to offer here in terms of this war on what could be a huge pandemic. Certainly is. And of course, that's, as I said, continuing on top of all the existing research programs. So on the COVID-19 side, our researchers have 6 current research programs. There are no guarantees on which treatment or vaccine is going to be effective. And we are drawing on quite a range of quite unique skills to find areas where we think that we have very unique opportunities, both in the short, medium and long term, to make a difference there. I won't talk about COVID-19 more right now. I'm happy to come back and talk about that question times. I think a lot of us have heard an awful lot about COVID-19. Today is really to hear about all of those other research programs that have continued thundering along in this new normal that we exist. Now, before passing on to our 4 researchers, I would like to just bring to life that statement that I said before, that Garvan's goal is to use all the information encoded in our genome to better predict, diagnose, treat and prevent major diseases that affect our society. And so that's a nice statement, but does it actually happen? What's the light on the hill, or the proof that all of this technology really will change the way we can, our health and our longevity? Well, the story that I want to tell you about is a young girl who now is 7 years old, but when she first joined a research program that was part of our clinical immunogenomics research consortium that led by Stu Taney and involved myself and quite and the Kinghorn Centre for Clinical Genomics here at Garvan, and the 2 major kids hospitals in Sydney. Carla was 1.5 years old, and at that time she'd come to Westmead Children's Hospital with a life-threatening overreaction of her immune system. In this case, her immune system wasn't overreacting against a virus, which is of course at the core of COVID-19. Her immune system was overreacting against her own body. In particular, it was attacking her own liver, and that was life-threatening, and it was attacking her joints. She had juvenile rheumatoid arthritis, and that was crippling. She had the best pediatricians you could ever find looking after her. They got on top of, by suppressing her whole immune system, they got on top of that liver autoimmunity, but the joint inflammation continued, and she started this continuing trial and error journey that everyone with rheumatoid arthritis goes on, of try this, one of the old poisons, methotrexate. If that doesn't work after a couple of months, then a biological, anti-TNF. And that's where she was when she joined our research program, and we sequenced her genome. And out of that came the answer. We had no idea, nor did her pediatricians, why her immune system was overreacting. It turned out it was a single change in one gene in her genome that leapt out. We already knew a great deal about it. It explained all her complicated syndromes, but better still, it matched her up with a medication that you rarely would use for rheumatoid arthritis, even though it's approved, because it only works in about 10% of people. But in Carla, all the science said it would essentially replace what was missing in a break in her immune system. And let me just share my screen, if I can figure out how to do that. This is multitasking, which is not my forte. But I have to share these photos in the background because to bring this to life, So here you can see Carla when she came into the Garvan last year. So Carla, back this time 3 years ago, went on to a targeted biological medication which completely transformed and suppressed her rheumatoid arthritis. And she's now 3 years out, no arthritis. She's just a wonderful, binging around little, a wonderful 7-year-old child, no more crippled, no more pain and journey. That's her with Carla's mum and the team that made the diagnosis. And you know, that's a short story, and I really just couldn't resist telling you that one because I think it illustrates how we can, through using all the information in the genome, start to personalise the way that people are treated, using all the science that we have and all of the teamwork that we can do between the laboratory and brilliant clinicians. And that's exactly the vision that drives us. And certainly for me, that's why I came to the Garvan and why I love to be at Garvan. It's a special time in medicine, and it's a very special place and unique place to be able to do that, recognising that it's a steep mountain to climb. Now, I really want to sincerely thank all of our supporters for their generous commitment, passion, and extensive contributions through 2020. It's really your support that's allowed us to be there for battling and helping people battle chronic diseases, some like ones we're going to hear about today, or like Carla's. And I'd like to encourage you to donate at the end of this seminar. We've been able to continue all our critical work in all of our major themes and expand into COVID-19 only due to the generosity of many donors in the community. It's one of the things that makes Garvan nimble and responsive to needs in the community. And if you're inspired today, please consider donating. Your support is vital for our work to continue. Now, I've probably gone over time, so I better hand back to our very charismatic compere, Mara Jean.
Our very bossy MC, I think, is the more appropriate description, Chris. Thank you very much, Chris. You touched on funding. What are your biggest concerns moving forward in terms of funding for medical research? We're no doubt approaching a major recession, potentially a global recession. How will medical research be funded into the future?
You know, I think the short answer is the same way. It'll depend on the same pillars that it has depended on for the last 10, 20 years. And those pillars are, if you look at Garvan, for all, you know, our efforts, it's 1/3 of that absolutely depends on philanthropy. And then 1/3 competitive peer-reviewed grants from government and other world bodies around the world, and then 1/3 from government, state and federal infrastructure money to support all the fabric of the building and the human resources and people and culture and the like. What I think, of course, is that all three of those sources of funding are going to be, really being hit by some big financial shockwaves over the next year. And so, of course, we're very, very concerned about how that will play out. We've, spent years recruiting some of the best and brightest from around the world. They're all under one roof, incredibly motivated, and they want to just crack on with solving these, very, very hard problems, which often take years of dedication. And it's going to depend on a consistency of support from the community. Having said that, I think one of the things that keeps certainly me buoyed, but I think each of the people you're going to hear from today is the engagement between the community and Garvan is quite extraordinary and unique. And I wish everyone could be in the building right now the way we have been able to, but particularly with the second wave unrolling, it's absolutely appropriate that we're doing this, sharing information, but not virus. And but it is one of the things that puts a spring in the step of every one of us is to hear from someone in the community who says, look, we really appreciate what you're doing, and here's a donation. And, we can only afford to give this much, but we're giving it to you because we really think what you're doing is important. And that sends a message that's so powerful.
It's very humbling, isn't it? Thanks, Chris. That's great. Before we introduce the first speaker, do you want to just give us a quick overview of the cancer theme at the Garvan Institute?
Yes, I'd be delighted to do that. So before I invite, you're going to hear from David Gallego-Ortega. And before I invite David to talk to you about his thing, I'd like to share a little bit about cancer. Our cancer researchers study the full gamut from rare and less common cancers, right through to cancers that run in families, different forms of sarcoma, pancreatic cancer, one of the most terrible forms of cancer still, one that took my uncle, and common cancers that have touched most of our families, extended families one way or another, things, and that come back still resisting treatment, like breast cancer, lung cancer, bowel cancer, bone cancer, and prostate cancer. And we have numerous clinical trials at different ranges that are really quite innovative, that bring together genomics with new medications that maybe have been shown to be safe or work in one common cancer, but the question is, can we apply them to other kinds of cancer? And that's really one of the strengths and one of the very exciting areas that's going on within Garvan, and its partnership with St. Vincent's, with the Clinical Trials Centre at University of Sydney, and with major cancer centres and hospitals around the country. So we're very much, the theme for cancer and for all of us is we don't try and do everything in our own backyard. It's impossible. We partner with everyone to get there as quickly and smartly as possible. And that's enough from me. Let's, if I can pass to David Gallego-Ortega.
Thanks, Chris. I'll just give David a very, very quick introduction. So Dr. David Gallego-Ortega leads the Tumour Development Group at Garvan. His group uses cutting-edge genomic technology and advanced tissue engineering to understand the molecular mechanisms of the cancer-to-immune cell communication, with the ultimate goal of developing next-generation cancer immunotherapies. David, great to have you with us. Welcome.
Thank you very much, Mara-Jean. Well, it's a pleasure for me to be here today, being part of this virtual seminar. I'm really enjoying it so far, and I'm sure it's a lot more to come. So let me share my screen. I think I'm in the right sharing. Can you see all that?
It's perfect. Thanks, David.
Thank you very much. Okay, so, well, good morning to everybody that is online. Today, I'm going to talk to you about cancer. So, as you all know, cancer is the second leading cause of death in our society. In 2019, only in Australia, 50,000 people died as a consequence of the big C, and another 145,000 new cases were diagnosed. This is equivalent to approximately one new case every 4 minutes. Currently, 2 out of 3 patients will survive cancer for at least 5 years. However, this greatly depends on the cancer type and the time of diagnosis. So in oncology, therapeutic recommendations are based on positive results on large clinical trials. However, this is a one-size-fits-all approach and remains ineffective in a significant proportion of patients. As a consequence, thousands of patients sacrifice their time and quality of life with ineffective and often toxic treatments. For this reason, precision medicine has arisen as the preferred approach for cancer medicine. Precision medicine uses specific information about a person's tumor to help make a diagnosis, plan treatment, or find out how well a treatment is working. For this reason, in vitro drug testing is a key component of personalized medicine. The problem is that conventional testing techniques based on cell cultures on a petri dish or using preclinical mouse models present fundamental defects and have failed to inform clinical decisions in the real world. So to provide a solution for that, my team here at Garvan has developed a personalized medicine platform based on a new invention, the Alten medical device. ALTEN combines advanced biomedical engineering with high-resolution molecular profiling technology available here at the Garvan Institute. The unique advantage of our innovative ALTEN device is its ability to preserve the whole tumor biopsy in a specialized gel. This medical gel is designed to mimic the tumor's natural environment and trick the cancer cells into behaving as if they were still inside the human body. As a result, ALTEN enables high-fidelity drug testing directly on the tumor biopsies of each patient, so we can accurately test which therapy works the best without ever having to expose any patient to the harmful treatments. So the process of ALTEN is simple. A tumor biopsy is excised and cut into small pieces. These pieces are introduced in the ALTEN medical device for gel encapsulation, forming what we call tumoroids. The encapsulated tumoroids are now challenged with multiple drugs and their combinations in vitro. And after a few days, those tumors are retrieved and analyzed using the latest pathology techniques and high-resolution genomic technology. With this approach, we're able to simultaneously analyze thousands of cells from each patient and classify them according to the drug sensitivity profile, informing clinical decisions real time. So, as a result, ALTEN is able to identify the best therapeutic strategy for each patient and potentially detects the early emergence of drug resistance in a very short period of time. So I would like to especially thank a couple of people in my team, Andrew and Laura, but specifically Andrew Lowe, that has been heavily involved with this project. And thank you all for your attention. I'm really looking forward to take some questions during the Q&A. Thank you.
Brilliant, David. Thank you so much. Chris, can I now invite you just to give us a quick overview of the genomics and epigenetics theme at Garvan?
Someone's turned off my video and I can't start it.
That's okay, Chris. We'll get that sorted for you in the background. There you are.
Too much technology for this old dog. Okay, David, thank you very much. That was, really wonderful and succinct. Now, the next theme that I want to introduce, and we're going to hear about from Joanna Achinger-Kawecka, is our genomics and epigenetics theme. Now, genomics, of course, is the study of our DNA, which is very complicated already, but as Joanna's going to tell us a bit more about, layered on top of our DNA is a set of marks that organize which genes are turned on and which ones are turned off to give a stable and heritable phenotype. And in fact, Garvan's researchers really lead the world in this space. So really, of course, it's that, just to link into cancer, cancer is partly a change in the DNA sequence, but what we know in almost every cancer is that one of the early steps in the evolution of a cancer cell going rogue is changes, large-scale changes in the way the DNA is marked and packaged. And that's probably enough for me, and we should hear from the real expert, Joanna.
Thanks, Chris. So Dr. Joanna Achinger-Kawecka is a Senior Research Officer in the Epigenetics Research Laboratory at Garvan. Her research focuses on investigating the role of the 3D epigenome in cancer, particularly in driving tumour progression, treatment resistance, driving tumour progression and treatment resistance, which is a major issue in prostate cancer. Joanna, over to you. Thank you.
Thank you. Okay. Hi, everyone. I'm honored to be able to present to you the research my team and I have been pursuing in order to understand the role of 3D DNA structure in prostate cancer treatment outcomes. So prostate cancer is the most commonly diagnosed non-skin cancer in Australian men, and with around 16,000 new cases every year. And to put that into perspective, that means that 1 in 9 men will be diagnosed with prostate cancer in their lifetime. And of these, more than 80% will be diagnosed with prostate cancer that needs hormones like testosterone to grow. And therefore, patients with advanced prostate cancer may receive hormone therapy, which will reduce the levels of these hormones in their body. And this can slow the growth of cancer or even shrink it. And currently, this can be done by either medical castration, so taking drugs that reduce testosterone levels, or surgical castration. But because these hormones are used by many other organs beside the prostate, hormone therapy comes with a wide range of life-altering side effects. And as our time is precious, even more when we have been diagnosed with cancer, we need to find better way to treat hormone-dependent prostate cancer with more targeted therapies. And this is why we are looking at the 3D structure of the DNA as a target to treat prostate cancer. So each of our cells contains 2 meters of DNA that is beautifully folded inside the microscopic cell nuclei. And even more amazingly, even though the DNA may look like it's completely randomly tangled up, the 3-dimensional arrangement of DNA is actually precisely folded in a very tightly regulated way. And it is this precise folding which makes our cells act the way they intend to be. And this is because inside that 3D DNA structure, the genes can touch or interact with different regulatory switches that tell the cell whether the gene should be expressed or turned on, or whether the gene should be silenced or turned off. So if the DNA is folded correctly, the gene can then be expressed, and the cell can do what it's meant to do. But if the DNA is not folded correctly, the gene cannot be expressed, and the cell may not be able to do what it's meant to do. And we have found that it is this incorrect folding of DNA inside the cell that can be a significant driver in cancer. So here we are comparing a normal cell on the left to a prostate cancer one by looking at how the DNA is folded inside it. And you can clearly see the difference between the 2 cells. So in normal prostate cell, there's this long ribbon, connecting ribbon between 2 regions on the DNA. And this connection is missing in the cancer cell. And this connection is actually located at a very important tumor suppressing gene located here. And because of it, this gene is not expressed in the cancer cell. So our goal now is to test if innovative treatments like epigenetic therapies can be used to target such 3D DNA changes, and if by reversing these changes, we can develop more effective treatments for cancer. So to sum up, we believe that our exciting research into the 3D DNA structure will reveal new therapeutic targets for more effective treatment for hormone-dependent prostate cancer, that will come with less side effects and therefore better quality of life after diagnosis. So on behalf of my team in the epigenetics labs, thank you very much for listening, and thank you for your support.
Brilliant, Joanna. Thank you so much. And what beautiful visuals you have. Chris, it's your turn to introduce us to the immunology and inflammation theme.
Thanks, Maritine, and thank you, Joanna. Very inspiring, and you made a complicated topic, I think, very accessible and bring out the interest. Okay, well, let's come to immunity and inflammation. Of course, it's the inflammatory and immune response that's at the core of COVID-19. It's at the core of so many diseases, and Garvan's researchers really are at the very leading edge in the world of using genomics to understand immunological diseases. And that's probably all I need to say before handing over for Marijine to introduce Elissa Deenick.
Thank you, Chris. That's great. Associate Professor Elissa Deenick is head of the Lymphocyte Signalling and Activation Laboratory at Garvan. Her research is focused on lymphocyte activation and differentiation and how this is controlled to ensure protection against infection while avoiding harmful immune responses like allergy and autoimmunity. Elissa, over to you.
Thank you. Yeah, so I don't just actually work on one disease, I work on multiple diseases. So that includes allergy. And we know that people can be allergic to many different things. So they can be allergic to peanuts, dogs, cats, pollen. And this can result in relatively mild things like hay fever or life-threatening conditions like anaphylaxis. I'm also working on autoimmune diseases, and this is a classic picture here of the rash that you can get with lupus, but there are many different autoimmune diseases like rheumatoid arthritis and autoimmunity that affects the thyroid like Graves' disease or Hashimoto's. And all of these cause a lot of problems for the patients. And I'm also working on people who suffer from severe infections such as pneumonia, which is an inflammation of the lungs, which can be caused by infections from viruses, bacteria, or even fungi. So how can I work on all of these diseases? What do they have in common? Well, really at the center of all these diseases is an immune cell called a B cell. And B cells are really important for protecting us against infection. And they do this by producing these little Y-shaped molecules that we call antibodies. So how do B cell responses work? Well, we actually have lots of different B cells in our body and they recognize different things. So if you get an infection, the B cells that recognize that infection will get activated, and then they need to expand so there's more of them, and then they can start pumping out large amounts of these little Y-shaped antibodies, which can then circulate all around the body through the bloodstream. And this allows them, wherever the bacteria or the virus that's infecting us is, to bind to the surface of that bacterial virus and neutralize it and help to eliminate it from our bodies. So that's how they're meant to work, but it can go wrong in some people. So in some people, they fail to make this really strong antibody response against infection, and that makes them susceptible to severe or kind of ongoing infections with like pneumonia. But that's not the only way B cells can go wrong. If, for example, we have a B cell that recognizes harmless environmental things like dogs or cats or peanuts, if these B cells get activated and start producing antibodies, that can actually result in allergies. Similarly, if we have B cells that recognize parts of our bodies, so bits of cells or bits of the thyroid, if these get activated and start producing antibodies, they can cause the disease associated with autoimmunity. So that's how we know that B cells go wrong and cause these problems. But why do B cells go wrong? So in my lab, we're trying to work out why this happens, and we're doing this by taking blood from patients, because the blood contains the B cells. So patients like Carla who have really severe forms of these immune diseases, allergy or autoimmunity or immunodeficiency. And then we can take out the B cells and test how they respond to different signals, so we can work out how these B cells have gone wrong. And we can also produce mouse models. So using these techniques and looking at some of these patients with really severe disease, we've been able to kind of identify some of the signals that the B cells are getting and that are going wrong and causing these diseases. And going forward, we hope that that will allow us to target some of these signals, to turn them up or turn them down, so that we can alleviate disease in these patients. So that's what we're working on in the future, and hopefully we'll be able to move to more of these common patients.
Just amazing, Elissa. Thank you so much. Such important work. Chris, The next theme you're going to introduce is the healthy aging theme.
Thank you, Elissa. Thank you, Mary-Jean. Well, you know, as we get older, medical research becomes more critical than ever because, of course, all the cells in our body start to degenerate for a variety of reasons. You know, every time we take a breath of air, that oxygen that keeps us alive is also damaging proteins and damaging our DNA. And so every breath is a step of degeneration and regeneration. Now, some of the cells that are, of course, really important and that we focus on in terms of diseases of aging are cells that control metabolism. For example, the cells that control our blood glucose, and so the whole problem of diabetes, of course, has been a major theme. The cells that allow our brain to move us about and remember things, and when they start to generate, of course, that's where diseases like Parkinson's disease rear up. And the cells that you might not think about so much, but are in one of the organs that we take for granted until we break our bone, but are actually one of the most special and wonderful organs of all, are the cells that make our bones. And Garvan's researchers are absolutely at the very leading edge of understanding the regeneration and degeneration of cells that make our bones, whether it's for cancer that homes to bone or for osteoporosis. And so one of our leading lights in that area is Michelle McDonald.
Thank you, Chris, and welcome, Michelle. Dr. Michelle McDonald leads the Bone Microenvironment Group at Garvan. Michelle has pioneered a new way of visualizing bone cells in real time, providing an exciting new approach to advancing biological understanding and treatments for diseases, including osteoporosis. Michelle, over to you.
Thank you very much, Mary-Jean. It's a real pleasure to be here this morning and to speak to you about the work that my group's been doing. Looking at osteoporosis has been mentioned, and the technique that we've developed in my lab, where we've developed an imaging window that allows us to visualize inside living bone tissue. That's allowed us to develop new insights that will improve treatments for osteoporosis. So as I'm sure many of you are well aware, osteoporosis is a disease where we see a loss of bone structure. As you can appreciate in these images at the bottom here, of the inside of the living bone tissue, of tissue compared to healthy bone, we see a loss of those bone structures. Osteoporosis affects over 2 million Australians. And as a result of the loss of bone in these patients, we see an increased risk of fracture, which affects quality of life. But more importantly, we've discovered now this also actually reduces survival in patients. And indeed, a quarter of our aged population will actually suffer from an osteoporotic fracture. Now, the bone loss that occurs in patients with osteoporosis is as a result of too much bone resorption by these cells called osteoclasts, shown down here, and also then a lack of new bone formation by osteoblasts. And because we see these changes in these bone cell behavior, we see a loss of bone structure as shown here. Now, my research team has been focusing on these cells here, the osteoclasts that resorb bone. And we know that they form through the fusion of these small cells into these large, what we call multinucleated cells, that then go on and undergo cell death when they've finished their job of resorbing bone. Now, there's a number of agents to treat osteoporosis that have targeted osteoclasts over the years, and a newer agent is called Prolia or denosumab. This is an anti-resorptive agent. It blocks the formation of these bone-resorbing osteoclasts, and in doing so, it really successfully increases bone mass and, importantly, reduces those fractures that impact survival. And as a result of the success of this drug in the clinic, it's now the #1 prescribed medication for osteoporosis for patients with osteoporosis. And as you can see, a large number of patients are prescribed this medication each year. However, it's become apparent now by tracking people over time that when patients come off their Prolia treatment, unfortunately, we're seeing a loss in their bone structure. So you see here a rapid loss in both these cohorts of patient in this grey area, which is following cessation or stopping their Prolia treatment. And the rate of loss of bone is much faster than the bone that they're accruing when they're on treatment. And as a result of this loss of bone structure, then we're seeing a really increase in the risk of fracture in these patients. Now, we do know that the cause for this is a rapid increase in bone resorption by osteoclasts that we'd previously stopped with treatment. So we really need to understand why this happens in order for us to really improve and prevent this loss of bone in these patients that have stopped treatment. So my group, as I mentioned, has developed this imaging technique that allows us to image deep inside bone tissue for the first time. And we can actually watch these osteoclasts as they move about and do their job resorbing bone in real time. And this movie here that I'm showing you on your left is of a red, you can see it as red osteoclast inside living bone tissue. And what you'll see is this cell will actually change shape and then break up into smaller cells and undergo a process we call fission. So it's the opposite of fusion. These cells are actually breaking up. And this is the first time that this has actually been documented before in the world. What we've also shown is that these cells not only undergo fission, but if you look here, this cell that does break up then actually migrates over here and fuses with this osteoclast. So they're undergoing cycles of fission and fusion, a process we've termed osteoclast recycling. And again, this is a completely new finding that we've discovered in our lab using this pioneering technique. So what we've demonstrated using our technique is that not only do these osteoclasts form through fusion to form these large resorbing cells, but they also undergo fission and cycles of refusion going through a process of osteoclast recycling. And this is a new biological finding. But more importantly, when we give Prolia in our models, we know that it blocks the fusion and formation of osteoclasts, but what we've also seen is that we see fission and an accumulation of these recycling osteoclasts. So that then when we take away the Prolia, as in modeling what's happening in the clinic when patients stop, these recycling osteoclasts that have accumulated are rapidly refusing to form these resorbing osteoclasts. And that's in fact what's driving this rebound bone loss that's happening in these patients. So through our imaging technique, we've developed a new understanding of osteoclast biology, this process of recycling, and that'll allow us to develop new ways to prevent bone loss and fractures in these patients that stop Prolia, and therefore allow the continued safe use of this agent in the clinic because it really protects patients from fractures and needs to be continued. So our team has developed new biological insights which will improve treatments, prevent fractures importantly, and importantly improve the quality of life and survival of patients with osteoporosis, which is a really common disease that affects our age population. So with that, I'd just like to thank a lot of donors who've supported this work as well as funding bodies. And thank you for your time. I look forward to questions.
Wonderful, Michelle. Thank you so much. Just welcoming everybody back to join me for the Q&A panel. We've got some fantastic questions coming through on the Q&A function. Before we go to those though, Michelle, is it true that osteoporosis can reduce life expectancy?
Absolutely. There's a lot of data that now supports the fact that once a patient has suffered from an osteoporotic fracture, it really dramatically increases their risk of death, unfortunately. And to the point where a patient who suffers a hip fracture in particular has the same risk or a reduction in survival and the same risk of death as if they had breast cancer. So it's really an issue. We don't understand the mechanism behind that at this point, but we know that it's a strong association. So understanding osteoporosis and preventing those fractures is really important, not only to quality of life, but survival of patients, absolutely.
Thank you. Thanks for that. And so for anybody in the audience who's concerned, would our recommendation be to speak to their GP about having a bone density scan?
Absolutely. So anybody over the age of 50 should really be keeping an eye on their bone density, see their GP, get a bone density scan and determine what's happening with their bones, and if they're considered osteoporotic, to get treatment as soon as possible and maintain that treatment to prevent the fracture. Don't wait until you fracture. That's our advice. Get that treatment in before the fracture happens.
And it sounds like stay on it.
And stay on it at this point. If you're on Prolia, do everything you can to stay on it, but we're working really hard to make sure that we have ways to prevent, if people do have to come off their Prolia, that we can stop that from happening.
Thanks. Thanks so much, Michelle.
Mm-hmm.
Okay, to the questions. Elissa, this one's for you. It's always great to learn something new, and this is the first time I think I've heard about B cells. Is there a simple explanation of how are T cells different to B cells? I thought T cells were part of the immune response, e.g. in the context of HIV.
Yeah, you're completely right. T cells are also part of the immune response. There's actually lots of different types of immune cells, and even in T cells, there's two different types. So the ones that are kind of destroyed and killed off in HIV are called CD4 T cells, and they're like the helper T cells. So they help all the other cells of the immune system to make the right response, which is why when you have HIV and AIDS and those cells get knocked out, you have such a problem fighting infection, because all those kind of helper T cells can no longer tell all the other immune cells what to do.
Thanks for that. Very good answer, very succinct. Now, this one's a clinical question, so I'm not sure that we will be able to address it. But Michelle, Prolia can be taken for how long and stopped when after 6 shots? So this might be somebody who's got a personal connection to this drug.
Sure. Yeah, I'm not a clinician, as Mara-Jean has pointed out. I'm a preclinical scientist. I work with a lot of clinicians, and there is the concern that we are seeing now following cessation of Prolia. So at this point, it's best to speak to your GP or specialist who's managing your treatment to see what the best plan of action is. If you do need to stop Prolia, then you need to look at an alternative to protect yourself from any bone loss, such as a bisphosphonate. But at this point, people have been well and have had no problem. The majority of patients have no problem on continued Prolia for up to 10 years or longer. So at this point, if there's no need to stop Prolia treatment, then continue taking it. If there's a concern and there's a need to stop, which doesn't happen in all patients by any means, then to speak to your GP about that. But hopefully all is well and you can maintain your Prolia treatment and it'll continue to protect you without any problems.
That's great. Thanks, Michelle. Chris, I'm going to throw this one to you. I'm curious to understand if exposure to viruses or bacteria, etc., in your childhood years is an important time for your immune system to learn what is an appropriate immune response.
Yeah, well, that's a great question, and Elissa will know as much about that as I do. But yes, there's now very, very compelling evidence that the immune system wires up on the encounter with even just the normal good bacteria in our guts, and so if those are not present, then the immune system is underdeveloped. There's always been correlative evidence. It's less watertight that if the immune system isn't busy fighting real infections, then it finds mischief. A bit like teenagers hanging around with nothing much to do. And that might be part of the reason for allergy, for example. But Elissa, you might talk more to that point if you're comfortable.
Yeah, I'd agree with Chris that, yeah, there's really good evidence that if you don't have good exposure in your youth, so for example, things like kids that grow up on farms and have lots of interaction with farm animals, where you get exposed to probably more bacteria than in a city clean house, have less allergies. I mean, there could also be other explanations for that. And also, something I kind of didn't cover about B cells is that they have a memory. So if you see an infection when you're a kid, let's say I see, a cold virus, then I can, make a memory response. My B cells will remember that down the road. So, and you'll be hopefully protected down the road. So it can train your immune system to know these are kind of dangerous things that can infect you. Remember what they are, so that next time you see them, you can make a response much more quickly and you won't get sick again.
Fantastic.
I suppose just to add one other point I was just thinking about is there's a very, very nice study that was done and now replicated in many other studies that in the case of peanut allergy, for example, it turns out that very early exposure by eating peanut-based food is a very powerful decrease in the incidence of developing peanut allergy. And so what that indicates is that the immune system, if it sees things very early on that we eat, it can learn to tolerate them. But if it sees peanut butter pasted on inflamed, rashy skin at age 2, it's more likely the immune system gets mixed up and thinks, oh, that must be a bacteria coming in through the broken skin, and that gives a 5-fold higher incidence of peanut allergy.
It's fascinating. That really contradicts what has been the most recent recommendation, doesn't it, in terms of exposure?
Yeah, those studies have really changed the recommendations now because it's absolutely rock solid, very large scale clinical trial data.
Fascinating, fascinating. Okay, I'm going to give this one to you, David. Your work is of obvious clinical relevance. When do you think your approach to breast cancer will make it to the clinic?
Yeah, so yeah, that's a very good question. I mean, we've done extensive preclinical assessment of the technology, and setting everything using preclinical mouse models and trying to identify the best treatment for, in this case, the mouse models. And now we are on the stage. Now we are launching the first clinical studies, and we're doing in parallel breast cancer and also colorectal cancer. And this will be answering a couple of questions on that, that the clinicians and the patients face basically every day, which is, what is the best treatment? There are some gray areas in those particular cancers where, and many others, but we need to start from somewhere, obviously, where, the clinicians have a moment where it's not clear what drug is going to benefit what patient. So we're trying to add information in there so the clinician can have a little bit more information to get a more pathological information to decide what patient. So the project for the clinical assessment of this is a 3-year project. So we're expecting to get data actually from 6 months in, but then after it's not completed, we cannot add the complete clinical value to the system.
Okay, fantastic. It's very exciting. Good luck. Elissa, another one for you, please. Lupus is not mainstream, but also more significant than many terrible diseases. Why has there not been more obvious progress for sufferers, and how would you describe the current state of play?
Yeah, look, it's hard. As an immunologist doing research on immunology, I often find it frustrating that there's often not as much press about these diseases as there are about, some other things, which I think is partly why, like, if you don't have as much funding, you can't make as much progress. Having said that, I think with the advent of genome sequencing and the ability to kind of look at these things, we are making progress on lupus, I think we're starting to understand that actually it's probably not one single disease, it's probably multiple diseases. And now I think the new technologies that we have are allowing us to kind of pull that apart and say, actually, maybe this is type 1 lupus, type 2 lupus, and they have different things that are driving them. And I think that in the next 5 to 10 years, we will actually see good progress as we start to give more targeted, personalized therapy to people with lupus and also other autoimmune diseases.
So a kind of a similar stratification approach as has been applied in cancer.
Yeah, exactly. Yeah, that's really what we need to do, because they, might look like similar symptoms, but actually what's going on underneath could be different. What's starting the disease in the first place.
Yeah, okay, really interesting. Joanna, a tricky one for you. Can genomics be done retrospectively on breast cancer? This is specific to breast cancer, but it could be prostate cancer as well, to know if a patient was given the most appropriate treatment for their cancer?
Yes, so this question is very much on the clinical side, I guess. But yes, we do collect tumor samples from patients when they go through treatment, and we frequently also collect the samples during the treatment, like for example biopsies, and then we can retrospectively look whether the patients were given optimal treatments or whether we could have changed something during the treatment. And this is very helpful because in the future we can then develop biomarkers to actually better stratify patients into correct treatments at the diagnosis, really, and avoid over-treatment or under-treatment.
Fantastic, thanks. Chris, before I go to the next question, I can't help but ask you, please, for a quick update on COVID-19. Obviously, we've seen a rapid change in Victoria in recent days and weeks. What are your thoughts? How is this going to play out?
Yeah, it's we're in the middle of a second wave. At the moment, it's Victoria. I suspect as the virus goes through 3 or 4 more cycles of 5-day infection cycles, we will see substantial outbreaks in other states, particularly here in New South Wales. At the moment, so much of the focus is on the quarantine side of things and maintaining borders and barriers. That's important. But what's really important is that R factor. If someone's unknowingly infected and going about their life in the community, do they infect one other person or do they infect 3? And that sounds like a small difference, but it's an exponential function. If it's 3, then those 3 people infect 9, who infect 27 after 3 cycles. After 4 cycles, you're talking about a very, very large cluster. And that starts to overwhelm the contact tracing. And so we all know we've got wonderful proof of exactly how to get that exponent on the R from 3 to 1 or below 1. We did it in April and May to flatten the curve spectacularly. Don't shake hands. We love shaking hands. We love going out to dinner. We love having a whole bunch of people over for a birthday party in our house. Don't do it. Even though we're allowed to do it, I think Gladys Berejiklian flagged yesterday, and I think and hope we'll hear in the next day or two that we don't want to go back into lockdown in New South Wales necessarily, but I think we need to go into something in between that, where simple steps that don't compromise business too much, but we already know how to do them, that just really decrease that exponent. So I'd certainly urge everyone to just be vigilant about that. The real problem in Victoria wasn't it was a small part of it was the leakage out of quarantine. As I understand it, may only have been 1 or 2 incidents of leakage. The real problem was that the community around Melbourne figured everything was back to normal. And so once it got out in the community, it was 1 goes to 3 goes to 72. And so anyhow, that's the short answer. Obviously, we're very hopeful about vaccines, but I spend a significant amount of every week keeping a close eye on good, high quality scientific data on how effective the vaccines are. At this stage, to be very blunt, it's not promising. I hope things change. One of the things that we're working very hard here is to bypass the need for a vaccine, or at least provide a stopgap in case vaccines take a lot longer or just simply don't work against this virus. And that involves using all of the knowledge about antibody genes and those B cells that Elissa Deenick, to sidestep all the complexity of our own immune system and do it all in the laboratory by gene technology. And that's where Professor Daniel Christ here at the Garvan is a world leader in that. And he's already produced a suite of fully human antibodies that seek out and neutralize the SARS-CoV-2 virus very effectively in a test tube. The real challenge is, can we make enough of that antibody to test if it's able to protect someone or treat someone who's been diagnosed with COVID-19? And that's really taking a lot of our attention and will take a lot of funding.
Thanks, Chris. It's sobering, but it sounds like there could be some good news on the horizon there with the antibodies. And now, apologies everyone, we do have a few questions that I've not managed to get to, some of them particularly of a clinical nature, so we would encourage you to raise those questions and concerns with your GP at your next visit. I'd like to give a warm thanks to our incredible speakers today. I think you guys did a phenomenal job with very, very limited time, so thank you for sharing your your incredible work with us and with our incredible audience. I must also acknowledge and thank Priyanka and Joseph, who are in the background making sure that today's session ran smoothly, or as smoothly as it could. They do a phenomenal job running these sessions, as well as many other important functions within the Institute. A special thanks to our incredible donors, our partners for the future, our advocates. Thank you for joining us today. There will be a questionnaire. We would sincerely value all of your feedback, your full and frank feedback. And thank you to everybody who has already made a donation this year. Your support is absolutely critical and transformational in the work that we do. And if you are inspired to donate as a result of today's seminar, please follow the link that will be up on the farewell screen. And thank you for your support and for your attention and time today. We're really grateful. Stay safe and well.